So I just want to acknowledge also my colleague in arms here, David Ingram, who's now working with the FDA. He was a graduate student in my lab at the time that he did this. It was a really fun project and we learned a lot. And so I want to give a lot of credit to him for doing this. So what is compost tea? We'll talk about, and what the potential for foodborne pathogen growth is in these using compost, and what are the ingredients. Some people add some additional things and what's the impact of that. Different kinds of methods for producing this, and again, balancing the safety with the benefits. So what it is, it can be either an active process or a passive process. And some people put additives in it and some people don't. You'll see some pictures of different kinds of apparatus as we go through here. People use these for all organic or various alternative farming practices as a method of creating either a soil drench, which is like a type of irrigation or a specialty application product, or as a foliar spray, or some people have larger systems and they make this compost tea extract to put into a fertigation line. And so they do this because they believe that it provides trace macro and micronutrients. They think that there's some evidence that it stimulates the plant's immune system as a type of a biocontrol for plant disease. They also think that some of the organisms that are growing in the tea are actually functional biocontrol type organisms, and it provides some particulate organic matter. So these are some few slides that talk about the health benefits for plants, and these were taken from Dr. Sherrell's work a number of years ago, where he looked at Pythium ultimum, which is a root pathogen that attacks seedlings, and the cucumber is the choice kind of plant to use for an assay for these. There are different kinds. He looked at teas made from kelp, which they added humic acids to. So those are these dark black blue circles. He also looked at ones where they added molasses formula to the formula for it, and so that's these red triangles. No additional nutrients, I should say. And then there was a water drench control and a heat treated compost tea. So they had a compost tea, but they heat treated it. So then they looked at the compost tea bacterial populations in colony forming units per mil used to drench the seeded pots. And then they looked on this other axis here was the percent of cucumber seedlings. So you see that there was quite a wide range of factors and responses to look at. But this kelp and humic acid was one that responded pretty well. But the cucumber seedlings sometimes did pretty well with the molasses formula, and sometimes not so well, as similar to these types of controls. So moving on to looking at, well, what was the quantity of the active cells? They stained these with some particular stains to look at that. And this was done with aerated compost tea that had molasses added to those teas. But they had some things that were excluded from these nutrients. And so they had a range of responses to these with the healthy cucumbers, again, on these axes here. So in some cases, they got good responses, in some cases not. So it was rather mixed review on whether it could control the Pythium. They also looked at powdery mildew, because this was way back a number of years before these things became of interest here in the United States. Europeans had looked at this sort of compost tea kind of thing, and they were particularly interested in controlling leaf mildews and molds on different types of plants. So they looked at this on roses. And in this case, all the compost teas used 0.3% molasses. Why do they use molasses? Molasses is a sugary compound. And if you remember what Margaret told you earlier today, she talked about how microbes, they need energy in order to reproduce. And the molasses provides the quick energy. So when you add molasses to a mixture, such as compost tea, you get an explosion in the population, but you're not just exploding one beneficial microorganisms, you're exploding the population of anything that can utilize that particular kind of compound. So it's an overall explosion in the microbial population. So there's no guarantee that the organisms that you don't want aren't going to be effective, or that you do want are going to be overly effective, as opposed to those that are just what we call cheaters, because they use some of the energy compounds that the beneficials really could use. So this is, they also compared the different kinds of compost tea starter materials and how they operated those compost teas. So some were operated as anaerobic materials, processes, and some were operated as aerobic processes. I'll show you some pictures of those. So basically these anaerobic processes in some cases don't do so well. CMC is just a particular brand of compost that had been of interest to this group. They use that. Anaerobic yard waste versus aerobic yard waste. So there's, again, you get a really mixed bag of responses out of these compost teas when looking at these types of leaflet inoculation kind of studies. They took some pictures and they showed that you can actually get these microbes to adhere to the cells on the leaves and actually maybe even get into the stomates here. So we were interested in, well, what is the potential for this? Is there a kind of a worst case situation that could happen out of this? I mean, you've got lots of competition because compost has a pretty active microbial population. But if you add the water that you need to make the tea, you provide aeration, and you have even very low levels of E. coli left in the compost. Or if you have salmonella, very low levels. Or another fecal indicator, not necessarily a pathogen, but just a good indicator of fecal contamination. What do we learn by this? If we start with low levels, is there enough competition to outcompete these? And that was one of the conjectures that a lot of people were making. So we used a 24-hour aerobic process, which is what a lot of people were saying. You could just use this aerobic process for overnight, or you could go for a longer period of time in an anaerobic situation. So we used some commercial nutrient solutions, 1 ounce per gallon, with molasses, bat guano, fish hydrolysate, humic acid, seaweed, seabird guano, kelp, all these different things in there. So we try this with different kinds of nutrient solutions. So what we see is that the inoculated compost in the 24-hour overnight compost tea, with looking at just the fecal coliforms and the E. coli, 2 different strains that we had, salmonella, and the anaerococcus. So the anaerococcus is just a general indicator for fecal contamination, but not necessarily a pathogen. So what do we see is that the inoculum level is the blue columns here. And then the nutrient solution A was one set, and that the populations here for fecal coliforms, they don't go up that much. But for in nutrient solution B, they really went up a lot. With nutrient solution A here, not a whole lot of difference between these, but there was a big increase over when we started from the inoculum that we started with. And with salmonella, it just seemed to explode with this nutrient solution A. So with none of these extracts, so you can hardly see this on this graph, there is a little black line down here near the axis. That's where there's no extra nutrients added to the compost tea. So in that case, even though we inoculated it, we don't see a lot of growth. So in the anaerobic situation, fecal coliforms, they like that situation. They grow quite well there. The salmonella did not do as well. And remember in the last set with this nutrient solution A, it goes way, way high. Here we hardly get any growth at all. It's just barely hanging on. Same thing with Enterococcus. So the anaerobic tea is not generally as supportive of the populations for this. And what's interesting is that this process that had been promoted, there were lots of different pieces of equipment that became available for people to easily use aeration. When in actual fact, when we see this, is you see that the growth from some of these is not so much, and it's more controllable by these nutrients and not so much by the anaerobic. So here's some of the aerobic methods that people use. They have these kind of containers, and there are some that are huge, much larger than this. But these are some of the different ones that David tried. And they have these permeable sort of net bags or socks in which the compost is loaded, and then the liquid is put into this some kind of a container. In some cases, you have an aerator that hangs on the side of the bucket. In this case, there's another type of aerator that goes into this one. And they want to get to dissolved oxygen levels that are at least above 6 parts per million. So that surely it is reaching something equivalent where it would be a pretty decent oxygenation level. An anaerobic method, not very much oxygen. It's a static situation. The compost is put into a permeable bag or net fixture. Water is added, and it's allowed to 8 or so days. And the liquid is used. 24-hour aerated brewer study. The heterotrophs were all in about gram negatives, were in general in these types of ranges. There were plenty of indigenous organisms present. Coliforms, again, can be relatively high, just general coliforms. E. coli can stay generally pretty low without these extra nutrients. Enterococci as well. This is some more additional study data, 24 hours. But the amendments, which we were asking him, which of these additives, because there were so many that people were promoting on the market to either use or not use, which ones should we look at? So we looked at this fish hydrolysate and all this sort of list of it, and we inoculated each of these separately with a very, very low level of pathogens. And what did we find? So we found here's the lineup of all these different compounds what would support the growth of E. coli 0157H7 in a 0.7% nutrient situation. So it's a really low level, but it's the same level for all of them. All of them went, this was what the inoculum level was, and they all went pretty high except for humic acid. And if you think about what we were talking about when we're looking for, well, how do we know something is good compost? You have this carbon-nitrogen ratio, as Margaret talked about, but we also know that one of the great products that comes out of a really good finished compost is a much higher level of humified material, and that gets to the humic acids. And so we see that when you have this humic acids, one, you have the acidic portion, and so that will tend to hold down some elements of a bacterial population that don't like acidic conditions particularly well. But it's the humic part, because this is a very complex type of molecule that forms in the soil over long periods of time. But that tends to hold it down, but it doesn't make it disappear. And so again, we come to the point of it's not pathogen-free, but there are some relative differences for these kinds of compounds. And so of all of these, I'd say if you had a choice, pick humic acid. It's the least offensive, but there's no guarantee that anything is going to knock out E. coli with these kinds of combinations. Same thing with this is a salmonella strain, and you see that the salmonella strain grows quite well. The humic acid also tends to stand out here as one that has the least encouraging of that population, least supportive of the population growth. So in conclusion, foodborne pathogens can propagate during aerobic tea production. And these commonly used additives, so you have this compost, which is supposed to be really good, has all these micronutrients, has a wealth of different kinds of microorganisms, is good enough to put into your soil by itself. So why do you think you need to add something to it? I can't address that, but people do, and they've tried all these different substances, none of which are guaranteed to knock out any of these pathogens. Several commercially available brewers can produce tea that contains E. coli and enterococci. So with that, again, we have a number of different boards. They had the National Organic Safety Board, had Compost Tea Task Force. You can look up on the NOP website and you can see all the details about what they considered. This was quite an in-depth look at it, and they came to some pretty reasonable conclusions about that. The additives are not recommended. It's the soluble carbon sources, like the molasses type things, but there are others, as you see from the list that were investigated here. And there are certainly a lot more area to look at in terms of the survival of these types of organisms and their transfer from soil onto fruits and vegetables. So with that, I will leave it at that.